In the very beginning of my postdoc project, so now I'm working to set up the OpenFE system, doing some silicon probes some acute recordings with silicon probes. So I think the best choice was present this ongoing data analysis from my PhD. So any comments and suggestions will be most welcome. So the title is High-Frequency Stimulation in CA1 Occludes Ketamine Effects in the Medial Prefrontal Cortex in Vivo. My supervisor was Professor João Pereira Leite, and my co-supervisor was Rodrigo Neves Ronce Pereira. So ketamine is a non-competitive antagonist of NMDA receptors. And it's a non-competitive because it binds in a different site of glutamate. So actually it binds in this site, and it's actually, even being an NMDA antagonist, the outcome of an acute treatment with ketamine is actually hyperexcitability, sorry. So this figure is a recent review from Halassa group, and they show in a very easy to follow way they trying to synthesize evidence from global to molecular levels. So here you can see finds in schizophrenia patients, and some experimental evidence from rodents and also with humans, linking NMDA receptors hypofunction and GABA dysfunctions. So, for example, positive symptoms, negative symptoms, and also cognitive deficits can be triggered by ketamine in humans. And also ablation of NR1 subunity is an obligatory subunity in NMDA. If you do that in mice, you can reproduce some of these positive, negative, and cognitive deficit symptoms. So also, there is, during the resting state in humans, if normal people, for example, they are treated with ketamine, it generates hyperconnectivity. And also, if you treat rodents with APV, that is another antagonist of NMDA, you can induce increased delta oscillations. Besides, aberrant gamma oscillations can be triggered by ketamine in rodents. And I think the most important here is that one idea from Lodge and other collaborators is that there may be from data from post-mortem, from tissue from patients with schizophrenia, they found loss of parvalbumin cells, parvalbumin positive cells in the hippocampus and the cortex. So they think these parvalbumin cells there is an imbalance between excitatory and inhibitory. So there is some hyperexcitability in the hippocampus, and this hyperexcitability can generate secondarily dysfunction in executive functions, for example, working memory, for example, or can generate hyperdopaminergic states. Because here you can see hippocampus has glutamatergic projections to nuclear accumbens, and nuclear accumbens inhibits ventropallido that inhibits VTA. So if this is too strong, this inhibition is also strong, and in turn, this becomes weaker, and dopaminergic becomes hyperdopaminergic, actually. And also there is another projection that's not represented here, that's VTA to the prefrontal cortex straight, right? And also these projections from hippocampus to prefrontal cortex, you can actually, there is these projections from the ventral hippocampus, but also from the intermediate hippocampus to the prefrontal cortex. And these projections are unidirectional, monosynaptic, and glutamatergic, right? So would ketamine be useful to model the hippocampus prefrontal cortex hyperconnectivity observed in some cases of schizophrenia? So this was a very, very basic question. And our aim was to examine and compare defects of systemic ketamine inject on the synaptic plasticity and the oscillatory patterns in CA1 medial prefrontal cortex pathway in urethane anesthetized animals. So another aim was to try, after characterize the ketamine effects, our aim was to try to attenuate the ketamine effects by prior LTP induction. So the rationale was because if you induce LTP, you're going to actually, if you stimulate, for example, imagine that this presynaptic terminal is coming from hippocampus. And this postsynaptic, for example, is located at the medial prefrontal cortex. If you stimulate in high frequency in CA1, you're going to promote glutamate release. So this glutamate is going to bind to AMPA receptors and is going to depolarize the membrane. And then this blockade with magnesium NMDA receptors is going to be, this magnesium is going to be expelled from the channel. Then, this high-frequency stimulation promotes calcium influx at the cell, and then it's going to promote synaptic plasticity events by intracellular cascades activation. So, what's going to happen if I induce long-term potentiation prior to ketamine effects? So, we test whether the prior induction of FTP is able to occlude the alterations induced by ketamine. So here is our two systems for acute recordings with animals anesthetized. We have here two Faraday cages and stimulators, analog to digital converters, stereotax, etc. So we anesthetize animals with urethane, and then we implanted electrodes in the medial prefrontal cortex prelimbic area, and also another recording electrode, a single wire, tungsten wire, was implanted in the intermediate hippocampus CA1, besides a bipolar electrode to do the stimulation and do the evoked potentials. So we, like Lesio did, we also apply paired pulse stimulation with 80 millisecond interval every 20 seconds to evoke response in the prefrontal cortex by applying electrical stimulation, CA1, right? And then after 90 minutes of baseline, we inject ketamine. So this was the first experiment. In the second experiment, we apply HFS twice at 30 and 60 minutes of baseline, and then inject ketamine or saline or vehicle. So this is just a panel to illustrate our system. The ground and reference was the same. Our reference was the ground. And that was over the parietal cortex. And we did the electrode recording was in the, like I said, in the prelimbic area, and the recording electrode in CA1 and the stimulation. And then we amplify the signal 100 times, and the filter, and then we sample the data with 10 kilohertz. And our stimulator was synchronized with the recording, So we could, like, at the very beginning of each window of 20 seconds of recording, at the very beginning we could take a zoom in and visualize the vocal potentials, right? So the results of the experiment 1. This is before ketamine, local field potentials filtered. Was collected in the prefrontal cortex. Here, the raw signal filtered in 1 to 2 hertz. And then here, filtered in gamma oscillation, 65 to 100 hertz. And then you can see that after ketamine, around 20 minutes after ketamine, you see that there is an increase in gamma amplitude oscillations. And then we notice that the ketamine group also display enhancement of delta oscillations. Remember that the animal is already anesthetized with urethane, so the animal most of the time already is oscillating in delta oscillations. But even though with a small dose of ketamine, I forgot to mention, I used 12.5 milligrams per kilo ketamine. And then you can see that the ketamine group, this is the x-axis time, baseline, ketamine injection or saline, delta, theta, beta, low gamma, and high gamma in the prefrontal cortex. So we got actually the ketamine-induced enhancement of delta oscillations and most prominently in the high gamma oscillations. So then we decided to do the modulation index analysis based on the TORCH work, 2007. And you can see here, before ketamine, I'm plotting here the amplitude of the high gamma filter signal, of the gamma envelope filter, and the delta, the low delta phase, right? So before ketamine and after ketamine. So both delta and gamma is being enhanced by ketamine, but high gamma is, the amplitude of high gamma is coupled to the delta phase. And you can actually quantify this in the medial prefrontal cortex before and after ketamine, in the medial prefrontal cortex and the hippocampus. And you can see that this modulation index is enhancing maybe 3-4 folds, is very robust. So ketamine increases phase amplitude coupling between delta and high gamma frequencies in both medial prefrontal cortex and CA1. Then we went to take a look in the evoked potentials, right? So this is how we measure the amplitude of the potentials. And you can see there I'm taking here a zoom in, okay? So we get 2 evoked potentials. And the second one is bigger than the first one, right? Because it's a paired pulse facilitation. This is a way, if you divide this one by this one, you can probe presynaptic plasticity, actually. So here you can see the normalized FPSP1 amplitude before ketamine and after ketamine. You can see that the animals treated with ketamine shows a 15% increase in the amplitude of the evoked potentials in the first potential. And also in the PPF, in the paired pulse facilitation, there is a decrease. So ketamine enhanced basal synaptic transmission and also disrupted presynaptic plasticity in CA1 medial prefrontal cortex pathway. Okay, see, yes? Decreases because increase the first pulse, but the second pulse is not together with them. Can you see? This is the second one. So when you divided the second one and per the first one, you got this decrease of the PPF. Any questions? No? So how about the whole of the long-term, the prior long-term potentiation? If you apply HFS before ketamine, what's going to happen? So this is the control group, saline and LTP saline. You can see baseline 30 minutes, and then I applied for the first time HFS. This is very robust phenomenon. You increase the amplitude, and then you apply again, and the amplitude becomes, actually you can see here like a saturation effect, because you apply it twice and there is no enhancement of that. And if you compare LTP saline versus LTP ketamine, the difference that I showed in the first experiment, I could not see such a robust effect that ketamine induced. So you can see a slightly higher amplitude in the ketamine, but it's not significant statistically. So the difference between control and ketamine-treated group disappeared in this case, or was attenuated by the prior LTP induction. And the most important that the PPF is we got almost an overlap between the two groups. So specifically in this case, the ketamine was, we couldn't see any effect of the ketamine if the LTP is induction before. So prior induction of LTP attenuates the short-term plasticity disruption induced by ketamine. So, and in the case of modulation index, we saw that if you compare, like, take a look just in the ketamine group during the baseline and the 30 minutes after ketamine, and then you take a look in the LTP ketamine group, you can see that actually there is an enhancement of the modulation index induced by ketamine, but is much lower, smaller than the ketamine without LTP, right? And this was very surprising for me because we got this difference only in the medial prefrontal cortex, but not in the hippocampus. And the hippocampus was the place that I actually stimulate. But the effect was only in the medial prefrontal cortex. So, main finds, experiment 1, ketamine enhancing delta and gamma oscillations in the medial prefrontal cortex. Ketamine increased phase amplitude coupling between delta and high gamma frequency in both medial prefrontal cortex CA1. Ketamine enhancing basal synaptic transmission and disrupt short-term plasticity in the CA1 medial prefrontal cortex pathway. Experiment 2, prior induction of LTP prevented the short-term plasticity disruption induced by ketamine, and the induction of LTP also precluded the ketamine effects on the delta phase, high gamma amplitude coupling in the medial prefrontal cortex, but not in CA1. So, from the literature, and discussing the results from the experiment 1, we could understand our data as maybe our data could be useful to understand and to discuss ketamine in a context of aberrant state of brain hyperconnectivity in schizophrenia. So the next experiments in freely moving animals, we could try to take advantage of this and try to understand how the circuits can be like changed during these ketamine effects and try to treat these animals. And also, regarding the prior LTP induction, we think that there are maybe two mechanisms that we could discuss. The first one is that actually if ketamine is an NMDA and is an antagonist of NMDA receptors, if I induce LTP before, I'm actually improving NMDA transmission, right? So if I do that, maybe I could try to avoid these ketamine effects in the NMDA. Also, it's already described from Therese J. group that if you induce long-term potentiation, if you do high-frequency stimulation in CA1, and you measure extracellular dopamine levels with the HPLC technique, you're going to see a very robust increase of dopamine, actually. And this fits very well with these models from Lodge and Grace. So if you do the high-frequency stimulation here, you are going to increase, actually, dopamine in the medial prefrontal cortex. So one interesting implication is that what would happen if we treated the animals with some dopaminergic agonist just after ketamine, for example? Maybe we could try to avoid this modulation index enhancement or this hyperconnectivity state. I don't know. And this is recent papers that the authors did deep brain stimulation in the ventral hippocampus and restored deficits in processing of auditory vocal potentials in a rodent developmental disruption model of schizophrenia. And this other one, they did the same. Both of them apply 130 hertz, and they don't discuss why. So we apply HFS that we know that induce long-term potentiation. We know that it enhances dopamine. We know that induce ZIF268 expression, specifically in the prefrontal cortex. So I think one important way to do deep brain stimulation is try to choose protocols that is biological relevant, or protocols that at least you know why you are choosing, right? So I would like to thank you very much, Professors João and Rodrigo, and also my lab colleagues. And I think you guys noticed that the organizing committee, they are actually all friends. There's like one family, right? So people is working with a great pleasure, and this is much more about the friendship and family than science. I think science is really important, but put people together and discuss and do things that they love, this is more important. Thank you very much.